A gene is a DNA sequence that encodes instructions for when and where to make a particular protein. But most of the DNA in our genome, more than 90%, is not made up of genes. The argument about the role of this apparently strange DNA has oscillated from one place to another. In the 1970s, it was thought that it was generally useless garbage. But in 2012, the ENCODE consortium, the DNA Elements Qt encyclopedia, right? He postulated that most of the DNA had some kind of activity. Earlier this year, a new analysis insisted again that it was just garbage. Although the debate was unleashing among researchers, viruses have used part of the non-coding DNA for their own purposes, to hijack our cell metabolism and promote its own replication. The results are reported in Science. ENCODE concluded that a large amount of non-coding DNA can be functional because much of it is transcribed into RNA, even if that RNA is not translated into proteins. There is evidence that some of these RNAs are functional, since they play a role in the control of other genes, although it is clear that not all RNAs play this role. Some of these RNAs, especially those that are relatively long, over 200 bases but not yet transformed into proteins, have been shown to be induced by a viral infection. A new work done in China has determined that viruses actually take advantage of one of these RNA molecules. The long non-coding RNA in question is manufactured by both mice and men. It is induced by a variety of viruses, and when it is eliminated, these viruses cannot replicate. It is not clear how all these different viruses activate the same gene at this point.